The intra-body nano network refers to a theorized and, according to a growing number of independent researchers, a partially or fully operational network of nanoscale devices deployed within the human body that are capable of communicating with each other and with external infrastructure using electromagnetic signals. The concept describes an internal biological internet, a mesh of nanoscale machines, self-assembling nanostructures, and biosensors operating within the bloodstream, nervous system, and tissues, exchanging data via radio frequency or terahertz band signals routed through the body's own conductive biological materials. The interbody nano network is understood by researchers in this field as a convergence of nanotechnology, brain-computer interface technology, microelectromechanical systems, and wireless communication, and is considered a central enabling mechanism of the Internet of Bodies and the broader transhumanist agenda. Critics argue that if operational, such a network would represent an unprecedented instrument of covert biosurveillance and biological control deployable against populations without their knowledge or consent. The scientific foundations for an intrabody nanonetwork are not speculative in isolation. Academic telecommunications researchers have published peer-reviewed work on the engineering requirements and feasibility of nanoscale communication networks operating within biological environments. A foundational paper by Ian F. Agildiz and colleagues, including the widely cited '08 Electromagnetic Wireless Nanosensor Networks, outlined theoretical architecture of nanonetworks operating in the terahertz frequency band, noting that graphene-based nanoantennas could serve as transceivers at this scale. Building on this, Agildiz's 2015 paper, Internet of Nano Things, extended the framework to describe how in-body nanoscale devices could interface with external internet infrastructure, effectively describing a biological extension of the Internet of Things operating inside the human body. These are not fringe publications. They appear in mainstream IEEE journals and form part of established academic literature on future communications engineering. What remains disputed and what independent researchers argue is already underway is whether such systems have been developed and deployed covertly, potentially using mass pharmaceutical interventions and aerosolized dispersal as delivery mechanisms. Key components: nanoscale devices. The functional units of the IBNN are understood to include nanobots, autonomous or semi-autonomous nanoscale machines capable of navigation, data collection, signal transmission within biological tissue, self-assembling nanostructures, structures that organize spontaneously from injected or inhaled precursor materials into functional devices. Researchers including Dr. Mahalgia and Mick Anderson have documented structures in post-injection blood samples that they argue are consistent with designed self-assembling nanotechnological systems. Graphene-based nanoantennas. Graphene oxide and reduced graphene oxide are identified in theoretical literature as the optimal materials for nanoscale electromagnetic transceiver construction at terahertz frequencies. Their alleged presence in COVID-era injections is considered by researchers such as Dr. Pablo Campra and Ricardo Delgado to be directly relevant to the IBNN deployment. Neural dust. Ultra-small wireless sensors originally developed at UC Berkeley, designed to be implanted through the nervous system and communicate via ultrasound. DARPA's neural dust program is a documented example of this technology. MEMS. Microelectromechanical systems. Microfabricated sensors and actuators that at their smallest scales approach the nano dimensions and are capable of detecting biological signals and transmitting data wirelessly. Communication infrastructure. For intra-body nano network to function, the nanoscale devices must be able to communicate both with each other intra-body and with external receivers or networks extra-body. Researchers have identified several mechanisms. Terahertz band communication. The frequency range most suited to nanoscale graphene transceivers, as established in peer-reviewed literature, the human body's biological materials are partially transparent at certain terahertz frequencies. Wireless Body Area Network, IEEE 802.15.6, the ratified IEEE standard specifically governing wireless communication in and around the human body, including in-body devices. This is formally published, publicly accessible standard that defines the technical architecture within which an in-body nano network would operate. Dr. Mahalchia is a physician and researcher who has conducted and documented extensive live blood analysis of blood samples from both vaccinated and unvaccinated using dark field microscopy. Her findings, published prolifically on her Substack, include anomalous filamentous and crystalline structures observed in live blood that were not present pre-2020, structures that appear to change form, self-organize, and grow over time, behavior consistent with self-assembly, but appear to be functional nanoscale devices embedded within or attached to red blood cells, the presence of similar structures in unvaccinated individuals, which she attributes to environmental exposure, potentially via aerosolized dispersal, or food and water contamination. Mahalchia has collaborated with other researchers to conduct spectroscopic analysis of these structures and argues that totality of findings is consistent with deployment of an intra-body nanonetwork through multiple vectors simultaneously. Mick Anderson, writing under pseudonym Corona2 Inspect, has produced some of the most technically detailed independent analysis of nanostructures found in COVID vaccine vials. His work cross-referenced observed structures against published scientific and patent literature on nanoelectronics, concluding that many observed formations are consistent with nanorouters, devices for routing electromagnetic signals within a nano network, nano antennas, graphene-based transceiver structures matching theoretical designs in the academic literature, nanorectenna arrays, devices capable of harvesting ambient electromagnetic energy to power in-body nano devices, potentially using 5G signals or ambient RF as power source, lipid nanoparticle delivery vehicles. Lipid nanoparticles identified as encapsulation mechanisms for delivering nanoelectronic payloads into cells. Delivery mechanisms. Researchers have identified several proposed vectors through which interbody nanonetworks components may be introduced into the human body. Pharmaceutical injections, particularly mRNA-based platforms using lipid nanoparticle delivery systems, which are capable of encapsulating and delivering nanoscale payloads directly into cells. Aerosolized dispersal via stratospheric aerosol injection and lower altitude spraying operation, potentially introducing smart dust class nanomaterials into the respiratory tract and bloodstream. Food and water supply. Some researchers, including Mahalchia, propose that nanomaterials consistent with interbody nanonetwork components have been introduced into the food and water supply, potentially via agricultural spraying or water treatment processes. Environmental ambient exposure. The ubiquity of graphene-based nanomaterials in manufactured products, combined with their stability and mobility in biological environments, may result in passive accumulation in human tissues over time. The broader agendas. Internet of Bodies. The IBNN is understood as a biological substrate for Internet of Bodies, the extension of Internet of Things connectivity to the human body itself. If operational at scale, the IBNN would enable continuous real-time biological data collection from large populations, feeding into centralized data infrastructure operated by state or corporate actors. The Wireless Body Area Network Standard IEEE 802.15.6 provides a formally specified short-range communication layer through which in-body devices would connect to this broader architecture.